The session A will be the Nobel Laureate Distinguished Plenary Structure lecture. So I'm going to introduce Dr. Gregg Semenza. We welcome him to our summit. He is a recipient of the 2019 Nobel Prize in Physiology and Medical Medicine. Dr. Semenza is a professor of genetic medicine at Johns Hopkins University School of Medicine. He was awarded the Nobel Prize for his groundbreaking discoveries of how cells sense oxygen and adapt to the change of the oxygen. So that really changed our view on understanding how cells adapt this, and this has been giving great impact in understanding many human diseases, including cancer, metabolic diseases, and blood eye diseases. Today, Dr. Semenza will deliver a lecture entitled Targeting Apoptotic Factors to Treat Cancer and Blinding Eye Diseases. Please join me in giving a welcome to Dr. Gregg Semenza. Please pull out your slide.
Thank you very much. It's a great pleasure to be back in North Carolina. I lived here for 2 years, about 40 years ago at the beginning of my career, when I was a health officer at Duke Children's Hospital. So it's a great pleasure to be back. Our work started about 30 years ago when we tried to answer a very simple question. We knew that the kidneys made a hormone called erythropoietin, and we knew that EPO was secreted into the bloodstream and in the bone marrow bound to progenitors of erythroid cells and stimulated their survival, differentiation, and proliferation. And we knew that when there's not enough oxygen delivery to the tissue, the kidneys make more EPO, so there are more red blood cells to carry more oxygen. But what we didn't know was how the kidneys could sense oxygen levels and respond to decreased oxygen availability with increased EPO production. And the first thing that we discovered was a short DNA sequence that's located downstream of the EPO gene. That's now known as the hypoxia response element. And this DNA sequence has the property that it can be taken out of the EPO gene, inserted into a heterologous gene, and the expression of that gene will be induced by low oxygen conditions. We then identified a protein that was present only in hypoxic cells that bound to this DNA sequence, and we called that hypoxia-inducible factor 1, or HIF1. We then used the binding of HIF1 to the DNA sequence as a way to purify the protein. And we found that it's composed of two different subunits that we called HIF1 alpha and HIF1 beta. So those two proteins come together and bind to the hypoxia response element, and then they recruit proteins called coactivators that serve as a bridge from HIF1 to the transcription initiation complex. And in addition, these coactivators are histone acetyl transferases that can modify the chromatin and allow the polymerase to access the DNA and transcribe it into RNA. Then in about 2000, the mechanism by which hypoxia regulates the abundance of HIF1 was discovered by several laboratories who found that there were two proline residues in HIF1 alpha that are subject to a modification called hydroxylation. So this occurs in the presence of oxygen. It utilizes oxygen and a metabolite called alpha-ketoglutarate. And these prolyl hydroxylase domain proteins insert one oxygen atom into HIF1 and the other oxygen atom is used to split alpha-ketoglutarate into CO2 and succinate, making this an irreversible reaction. The hydroxylation of HIF-1 alpha, which occurs in the presence of oxygen, allows the binding of the von Hippel-Lindau protein, VHL. And when VHL binds to HIF-1 alpha, it recruits a ubiquitin protein ligase, and HIF-1 alpha is ubiquitinated and degraded in the presence of oxygen. We also discovered a protein that interacts with HIF-1 alpha that we called FIH1 for factor inhibiting HIF-1 because it blocks the activity of the protein. And it turned out that FIH1 also modifies HIF-1 alpha under normoxic conditions. And it uses the same biochemistry as the prolyl hydroxylases, but it inserts an oxygen into an asparagine residue in the transactivation domain. And this hydroxylation prevents the interaction with the coactivators. So you can see that under normoxic condition, the protein is both inactivated and degraded. And so you can appreciate that this provides a mechanism to directly transduce changes in oxygen availability to the nucleus as changes in HIF activity. So it's a very beautiful homeostatic system. Again, under hypoxic conditions, these modifications do not occur, and the protein can rapidly accumulate within hypoxic cells, dimerize with HIF-1 beta, and activate target gene expression. Early on, we knocked out the gene encoding HIF-1 alpha in mouse embryonic stem cells, and we found that mouse embryos that were homozygous for the mutant allele arrested in their development around embryonic day 8.5 and died by day 10.5 with malformations of the heart, the blood vessels, and decreased red blood cell formation. So all three components of the circulatory system are dependent on HIF-1 for their normal development. Well, it turned out that the situation was a little more complicated because HIF-1 alpha has two siblings, HIF-2 alpha and HIF-3 alpha. These are the products of distinct genes, but they're structurally and functionally similar to HIF-1 alpha in that they dimerize with HIF-1 beta and activate transcription. But whereas HIF-1 alpha is present in all nucleated cells of all animal species, HIF-2 alpha and HIF-3 alpha are only expressed in certain cells and only in vertebrate species. So they're evolutionary add-ons. So that's the basic history of the discovery of the transcription factors. And now I'd like to tell you about our recent work targeting these transcription factors therapeutically as a way of treating cancer. And so we have 3 different platforms that have led to a discovery of inhibitors, and I'll tell you about 2 of the platforms today. So the first one involves this small molecule that we call S9N. Okay, so just this as a step back, I told you that we discovered EPO as being regulated by HIF-1. Now we know of over 10,000 RNAs that are regulated in exactly the same way. HIF-1 or HIF-2 binds to the gene, activates transcription. And you can see some of these genes serve to increase oxygen delivery, like EPO is a response to systemic hypoxia, but there's also local tissue hypoxia when, for example, the coronary arteries become narrowed and the flow of blood to the heart is impaired. Then there are local responses which involve a new blood vessel formation. And so vascular endothelial growth factor, or VEGF, is an example of a protein that stimulates local tissue oxygen delivery. And then regardless of whether the hypoxia is systemic or local, individual cells must adapt to decreased oxygen availability, and the classic metabolic adaptation is the switch from oxidative to glycolytic metabolism that's mediated by HIF1. So unfortunately, this beautiful system is hijacked by cancer cells. On the right is a immunohistochemical assay of a typical aggressive cancer. You can see these clear white areas labeled necrosis. Those are areas where the cancer cells have died. And they've died because there is no oxygen available to them. All the oxygen has been consumed by the cells between them and the blood vessel where the oxygen is delivered to the tissue. And you can see these brown stained nuclei, and they've been stained with an antibody against HIF1 alpha. And you can see that these are the viable cells that are the furthest away from the blood vessels. So these cells can survive the very low oxygen conditions, and their adaptation also allows them to survive radiation therapy, chemotherapy, any therapy that patients are administered. And it's these cells that have the greatest propensity to invade and metastasize and kill the patient. And until recently, there were no drugs that targeted these cells. So that was our goal, to develop inhibitors of the hypoxia-inducible factors that would target these hypoxic cells. Well, you may know that the first in-class HIF inhibitor is a drug called belzutifan, and its structure is shown at the top right. This compound binds selectively to HIF2 only. And it was approved by the US FDA in 2023 for the treatment of renal cell carcinoma. And it's had subsequent approvals for several other rare cancer types. It was developed by a startup company in Texas that Merck bought for $2 billion. And this is a pretty good investment because you can see that last year their sales have continued to rise every year. So this is very important because it provides proof of principle that these transcription factors are druggable. Because for 20 years I listened to people tell me how you couldn't inhibit a transcription factor. And so this drug binds to HIF2 alpha and blocks its ability to interact with HIF1 beta. So a functional transcription factor cannot form. Well, we know from extensive preclinical studies that both HIF1 and HIF2 must be targeted for effective cancer therapy in most cancers. And this is because some genes are regulated by HIF1, some are regulated by HIF2, and some are regulated by both HIF1 and HIF2. So in order to block the hypoxia response, you have to block both HIF1 and HIF2. HIF3 is not expressed in many tissues, and its role is not well understood. So there haven't been any subsequent belzutifan approvals for more common cancers. And I believe this is because renal cell carcinoma is an outlier in that the disease progression is driven mostly by HIF2. And so inhibiting just HIF2 has a beneficial effect. But as I mentioned, in most cancers, immunohistochemistry studies have shown that high HIF1 alpha in the diagnostic tumor biopsy identifies patients who are at increased risk of dying of their cancer compared to patients with low HIF1 alpha in the primary tumor biopsy. So we believe there's a major unmet clinical need for a dual HIF1, HIF2 inhibitor. So I'm going to tell you about S9N, which is a first-in-class dual HIF inhibitor that inhibits tumor growth and vascularization. It also sensitizes cancers to immunotherapy. It's active with once daily oral administration. The head-to-head test that we've done is superior to a HIF2 selective inhibitor, and it seems to have an excellent safety profile. And we believe it will address a major unmet clinical need. So how did we develop S9N? We started with a computer-aided drug design strategy. We collaborated with a laboratory at the University of Maryland that over 10 years developed an algorithm that can take a crystal structure of a protein and predict ligand binding sites. So this program predicted 3 candidate binding sites and gave us a prioritized list of candidate ligands, which we then purchased and tested in cell-based screening assays to identify compounds that could inhibit both HIF1 and HIF2. And we had 2 hits from our primary screen that we then did a tertiary screen looking at hundreds of analogs and then testing the best candidates in tumor models. And so we ended up with S9N as our preclinical candidate. And you'll see it has quite favorable properties in mouse models. This is what it looks like. It's a beautiful symmetrical molecule. So what does it do? It binds to a domain in HIF1 alpha and HIF2 alpha that's highly conserved. 90% amino acid similarity, explaining why it can bind to both HIF1 alpha and HIF2 alpha. It's a domain called a basic helix loop helix domain, which I don't believe has ever been targeted in a transcription factor by a small molecule. The binding of S9N prevents the interaction of the alpha subunits with the beta subunit, and then triggers the degradation of the alpha subunits, so they're completely destroyed. This has, of course, a profound effect on the expression of HIF target genes and tumor growth and vascularization. This is a particularly horrible cancer called glioblastoma multiforme. The patients who have this cancer require brain surgery, then they have irradiation to the brain, then they're treated with cytotoxic chemotherapy, and the end result is they do not survive more than 15 months. So there's really no effective therapy for this condition. And the HIF1 alpha levels are very high in these tumors. And so in a mouse model, we established the tumor and started treatment with S9N once a day by oral administration. And you can see as soon as we started treatment, the growth of the tumor stopped. This is another model. This is a genetically engineered mouse model of breast cancer in mice. So the mice have been engineered to express an oncogene in breast epithelial cells. And in this model, by day 53, the female mice start to have palpable mammary tumors. And so we started treatment on day 53, either with S9N or belzutifan or vehicle. And this shows a Kaplan-Meier curve of the survival of the animals. And you can see that by day 94, all of the vehicle-treated animals have died. On that day, none of the animals treated with S9N had palpable tumors. And you can see they eventually develop tumors, but their survival is greatly increased. And in contrast, belzutifan had no significant effect on survival of the mice. I told you that S9N has very powerful effects in combination with immunotherapy. So here we chose 5 different mouse tumor models, and we chose them because they're all known to be resistant to immunotherapy. And we established the tumors and treated the animals either with immune checkpoint blockade alone, either anti-PD-1 or anti-CTLA-4 therapy, or we treated them with immunotherapy plus S9N. And you can see that the sum total, 1 out of 25 mice treated with immunotherapy alone had a complete response, which we defined as eradication of the tumor, no tumor recurrence after discontinuing all therapy, and then we could re-inject cancer cells and no tumor would form. So 4%, that's not far off from what is observed in patients. In general, these drugs don't work because the cancer has many ways of evading the immune system, and the therapy only activates one of them. But you can see when we treated with S9N in a combination therapy, we had an over 10-fold increase in complete response. And we know the mechanism for this because we know that cancers exclude T cells and NK cells from the tumors. And if there are no T cells in the tumors, the immunotherapy cannot work. And when we inhibit HIF1, we increase the recruitment of T cells and natural killer cells to the tumor. And this provides the mechanism for this dramatically increased response to immune checkpoint blockade. So obviously, if we could achieve this in the clinic, it would save thousands of lives every year. Is S9N safe to administer? So we treated animals for 40 days with twice daily oral administration, so twice the therapeutic dose. We saw no change in the appearance, behavior, or body weight of the animals, no change in blood chemistries on day 40, meaning normal liver and kidney function, no change in platelets or white blood cell counts on day 40, indicating normal bone marrow function, and no change in histology across 8 organs. The only thing we noticed was a small decrease in the hematocrit, which is an on-target effect of inhibiting HIF2 and thereby inhibiting EPO production. So anemia is the most common adverse effect of belzutifan. Okay, so what are the targets that we would like to treat with this novel therapy? Well, we like to target cancers for which there's no effective therapies. These include triple-negative breast cancer, glioblastoma multiforme, metastatic colorectal, recurrent head and neck, and advanced lung cancer. And we've tested the drug in mouse models of each of these and seen therapeutic effects. These are all very aggressive cancers with a short progression-free survival, which means that if there's efficacy, it can be detected in a relatively short period, 6 to 12 months. And in addition, glioblastoma is an orphan disease, and so clinical trials focused on that cancer are streamlined. Well, I told you about belzutifan. There are 3 other drugs that act just like belzutifan that are in clinical trials, and a small interfering RNA that targets HIF2 alpha that's in phase 1. There's a drug in phase 2 that inhibits HIF1 beta, but HIF1 beta is not just a subunit of the HIFs, it's a common subunit for a number of other transcription factor families. So that is a much less specific approach. It's much more likely to have other adverse effects. And that's why we never pursued that approach. There are no HIF1 alpha selective inhibitors in clinical trials and no dual HIF1, HIF2 inhibitors in clinical trials. And you can see that there are other compounds that are in development, but for none of these is there published evidence of anti-tumor activity of the sort that I've shown you. Well, what are other treatment strategies that are being developed to try to overcome the block to immune therapy? Well, one is to develop these bispecific antibodies that recognize both PD-1 and vascular endothelial growth factor. So how does a HIF inhibitor plus immunotherapy production and thereby inhibit the recruitment of immunosuppressive myeloid cells? We've demonstrated that in our models. But probably the greatest concern of these bispecific antibodies is that by inhibiting VEGF, there'll be an inhibition of tumor angiogenesis, which will cause hypoxia, which will increase HIF activity in the tumors. And that's never a good idea. So actually, I don't think these drugs should be used unless they're given in combination with a HIF inhibitor. This is why VEGF antibody therapy is not effective in cancer. So I mentioned that belzutifan and inhibiting HIF2 alone seems to be fairly effective for renal cell carcinoma. Unfortunately, the most recent belzutifan trial as first-line therapy in a 3-drug cocktail failed, but it's still been approved for second-line therapy. But as I mentioned, there are all these more common cancers where both HIF1 and HIF2 are known to play a role. And as I mentioned, we've tested S9N in mouse models of each one of these and found it to be effective either as a monotherapy or in combination with immune checkpoint blockade. So we developed, started a company called HIF Therapeutics to target the HIFs for treatment both of cancer and ocular disease, as you'll hear about. And I started this in combination with Gregg L, who's a retinal surgeon at Johns Hopkins. And we've done the drug discovery work in my lab and the cancer models. And in his lab, they use multiple mouse models for blinding eye diseases, as you'll see in a moment. And then most recently, at the beginning of this year, we were very fortunate to bring on Julia Yang, who many of you have met already. And she's been helping us to try to develop our business. Okay, so I'm just going to take a short drink here and go on to the second part if there's no questions. Okay. So just a little bit. Obviously, we're at the stage now of trying to raise funds to do the IND enabling studies by a contract research organization. And this is the summary of what I've already told you. I'll just mention that 10 million people die in the world every year because there's no effective therapy for their cancers. And we think HIF1 plays a big role in many of those cancers. Okay, so I mentioned that my colleague Gregg L has taken some of the drugs that we've discovered and tested them in the mouse models of blinding eye diseases. This includes diabetic eye disease and then age-related macular degeneration. And these diseases are the major causes of adult onset blindness. It involves hundreds of millions of people worldwide. The principal therapies are all targeted to VEGF. So these diseases involve a pathological increase in vascularization of the retina. And so VEGF can inhibit that vascularization in some patients. And so these drugs are now the standard of care. But as you'll see, they have limited effectiveness. Not only that, but they require a needle being stuck into your eye, which is not a pleasant thing. And that needs to occur every few months. Again, only about half of the patients or less respond to these drugs. And those who do respond, they usually lose the effectiveness of the treatment by 5 years out. And there really haven't been any new therapeutic approaches in the last 20 years. So this is a HIF inhibitor that we developed through another platform. It also inhibits both HIF1 and HIF2. It's very effective in mouse models of these ocular diseases, and even when tested head-to-head with anti-VEGF therapies. And probably the most exciting part of this is because they're small molecules, they can be administered orally or by eye drops or by injection, rather than biologics, which must be injected into the eye. And not only that, because they're targeting HIF1, HIF1 not only controls VEGF production, but it controls the expression of other angiogenic growth factors. And we think that VEGF inhibitors are ineffective in the majority of patients because other angiogenic factors are also being expressed, and the therapies do not target them, whereas inhibiting HIF1 does. The other important thing is that the anti-VEGF therapies, they completely eliminate VEGF activity. And for some cells, VEGF is a survival factor. When we inhibit the HIFs, we don't affect basal VEGF expression. We only affect the pathological increase in VEGF expression. And so we think that using HIF inhibitors will be safer than using VEGF inhibitors in the long range. So this slide shows all these different angiogenic growth factors that are produced in the eye in response to hypoxia and ischemia. Some of these drugs target both VEGF and a second angiogenic factor, but none of them target all of these factors other than HIF inhibitors. So we think that certainly patients will appreciate eye drops as opposed to intraocular injections. It should be mentioned that, of course, to get an intraocular injection, a patient has to go to a healthcare facility and be injected by a physician, whereas eye drops can be taken at home. And so we think that this is going to greatly expand the market because many people do not seek therapy because it's either too expensive or too inconvenient. This is a mouse model of diabetic retinopathy looking at the development of blood vessels, and on this the slide on the right shows the retina kind of splayed open, and the green represents new blood vessel formation. And you can see that the animals treated with this HIF inhibitor 32134D had an 85% reduction in new blood vessel growth. So the drug is very effective in blocking new blood vessel growth in mouse models. And you can see that Ockrid's lab has published several papers on these. In addition to proliferative diabetic retinopathy, there's also diabetic macular edema, where the part of the retina where the sharpest vision occurs swells due to fluid accumulation. And you can see in these diabetic mice at the far right that the animals treated with 32134D have a great decrease in the extravasation of dye from the retinal blood vessels. So the leakiness that occurs with diabetes in the eye blood vessels is blocked by treatment with a HIF inhibitor. Here, this is comparing an anti-VEGF inhibitor called aflibercept or Eylea with our HIF inhibitor. And you can see for both of them, there's a dose-dependent inhibition of blood vessel formation. This is a model where the animals are treated with a laser to stimulate blood vessel formation. And then 3 days after the laser, they're given an injection of either the VEGF inhibitor or the HIF inhibitor. And here you can see that a dose of the VEGF inhibitor, aflibercept, that doesn't have a significant inhibitory effect on tumor growth, when combined with a very low level of the HIF inhibitor, results in a therapeutic response. So we think that this drug will be useful in combination with the VEGF inhibitors and may improve their efficacy in people who still want to get an eye injection, I guess. We're hopeful that it'll work as a monotherapy, but certainly this is probably a good starting point. Okay, I told you that the thing that we'd most like to develop is a topical formulation. And so this is a study where we administered 32134D topically 3 days after the laser, 3 times a day for 3 days. So the mouse got 9 drops in its eye, and you can see, and this was the very first time we tried this, so this is by no means optimized. And you can see the significant reduction in neovascularization that occurred in the animals that received the topical 32134D. Again, this is the strong point of a small molecule, its ability to diffuse through the tissue. Akhbar's lab also gave very high doses of the drug by intravitreal injection and then followed the animals for long periods of time with electroretinograms to look at the retinal function and histology. And he found no toxic effects of these drugs even at 50 times the therapeutic dose. So this drug is remarkably safe to use in the eye, we believe, at least in mice. Okay, so as I mentioned, there really haven't been any strategies other VEGF inhibition in the last 25 years. This is a very different and we believe more effective therapy because it will target multiple angiogenic factors and will target the pathological increase in the expression of these factors in the retina. As I mentioned, the use of topical administration should greatly expand the market and make this kind of treatment available to many patients who currently are not treated due to expense or unavailability of the therapy. So here's the summary. We're very excited about the further development of this drug. And as in the case of S9N, we filed patents regarding 32134D. We're also working on liposomal nanoparticles as a strategy for delivery, both topically and by injection. And so that's our company, and we hope you like our innovation, and we would certainly like your investment. Thank you very much.
So let's stay down so we can have questions from our audience.
Hi, Dr. Semenza. Congratulations again on the groundbreaking, discoveries in the HIF pathway. Very fascinating works on the new initiatives you're working on these 2 programs. My question more focusing on oncology implications there. I'm just curious, do you have a particular biomarker strategy to identify certain populations that could benefit from the drug? The other question is related to the safety. Do you think about potentially antibody conjugate or peptide conjugate to help you to enrich to a certain tumor tissues versus the normal tissue?
Yeah, those are great questions. The microscope is great.
Actually, you have my microphone.
We're good? Okay.
Yes,
All right, great. Tell me your first question again. Biomarkers, yeah. So there's 2 things about that. First, the HIF-1 alpha expression in tumors tends to be very heterogeneous. And so the question is, will you get an accurate readout, for example, by immunohistochemistry or will you miss some cases? The other important issue is that we know that many cancer therapies, one of their consequences is they induce defective. So as I mentioned, for example, angiogenesis inhibitors cause hypoxia, they induce decline. So we believe that a lot of cancer therapies, not just immunotherapy, will be more effective when they're combined with a HIF inhibitor. So at least to start, I'm not particularly in favor of biomarker limitation. I think it'll be very useful as a post hoc analysis, but not. I don't think we should bias our patient population quite yet. What was the second one?
The second question is about whether you're interested in
Yeah.
anti-conjugate.
Right. So, as you know, strategies that increase selectivity reduce the overall delivery. And we know that cancer is a systemic disease, and you've got to get your therapy everywhere. And again, this is why I feel there's still a strength to small molecules, right, that are dissolved in the body water, right, and go everywhere, because the cancer cells go everywhere. So that's certainly a potential strategy, but my own bias is to treat a systemic disease with small molecules.
Thanks for such a wonderful talk and the beautiful data you showed. I actually also have two questions. The first one, I was thinking for the HIF pathway. In our body, maybe we have the hypoxic condition, which is a pathologic condition. Also, we may have some the physiological hypoxic condition. How do you think about those kind of HIF pathway in this one? This condition.
Yeah. So, we know, for example, that the HIFs play an important role, a protective role in ischemic cardiovascular disease. And so you may need to exclude certain patients with severe ischemic cardiovascular disease. Now, remember that belzutifan has gone through clinical trials. I've looked at their exclusions, and I don't think we would need to have any additional exclusions from what were used in the belzutifan trials. And, the only things, well, I should say the highest incident adverse effects in those trials, as I mentioned, anemia on target, and then interestingly, hypoxia. And the hypoxia is not from the anemia, okay? And we knew that HIF2 plays an important role in the control of breathing in mice. So we asked a respiratory physiology laboratory at the University of Chicago to give vehicle belzutifan or S9N to mice and measure their breathing. And the animals treated with belzutifan had periods of apnea where they stopped breathing. The vehicle-treated mice and the mice treated with S9N did not. And we were not surprised about that because our studies indicated that the control of breathing requires a balance between HIF1 alpha and HIF2 alpha. And when that balance is...
How many genes got affected between the case and control after giving the treatment? I really want to see how many genes got up and down regulated because it will help us to design further drugs targeting those genes.
Yeah, so we did RNA-seq on a tumor cell line in culture, right, where we exposed the cells to 20% or 1% oxygen for 24 hours. And we see hundreds of genes go up and hundreds of genes go down. And there were hundreds of genes affected by S9N. And I told you that we had 2 hits in our original screen. And the other hit is at a different site. But we also, we carried that quite far forward. And the overlap between the 2 drugs on the RNA-seq was very striking. In contrast, belzutifan was off in left field. And some of those, the majority of genes inhibited by belzutifan were not induced by hypoxia. I don't know what they are, but it was quite striking, the difference. Whereas our two inhibitors had remarkable overlap in the targets, even though they target different places on HIF1 and HIF2.
Thank you very much.